anatomists and physiologists. This video is part 2 of our reproductive series. This is all about meiosis. In the last video, we talked all about the anatomy of male and female reproductive systems, but this time we're going to talk about the process of how those gametes, those sperm cells and those egg cells, are actually produced. So the process of gamete formation happens by this guy called meiosis. And meiosis is cell division. And we've learned about cell division before, but that's called mitosis. So I'm going to write meiosis does not equal mitosis. Similar but different processes. In the process of mitosis that we learned about in Bio 430, we learned that one parent cell would produce then two identical daughter cells. Those daughter cells were identical. But the process of meiosis is a little bit different because we form gametes. Meiosis is actually two processes, meiosis 1 and meiosis 2, that results in gametes, sperm cells or egg cells. And a lot of this comes from the idea that when we combine sperm and egg, that means that those two cells come together, and that can mean that we may have, think about the amount of chromosomes, the amount of copies of chromosomes that we have. And so a normal cell, like a body cell, a somatic cell is what we call diploid, or 2N, which means that it has two copies of chromosomes. So that somatic cell would be like 2N, two copies of chromosomes. Gametes are haploid. Sperm cells and egg cells, they're haploid, which means that they have half of the number of chromosomes, 1N, haploid. And that's because when sperm meets egg, then sperm plus egg, if they're each 1N, they would become two copies of chromosomes. So we have one copy of chromosomes from our mom, one copy of chromosomes from our dad. And chromosomes, if we remember, are just like those bits of DNA that get wound up really tight. We as humans have 23 pairs of chromosomes for a total of 46, because we have two pairs of them, one from mom, one from dad. And then we divide those again to form gametes. So just as a reminder, chromosomes are bits of DNA, and we oftentimes draw them like this. Chromosomes are kind of like in a boop. Little bits of DNA with a little center thing there in the middle. And once that chromosome multiplies rather, copies itself, then we get a duplicated chromosome, which kind of looks like this. One chromosome, and then its copy. And then again, that center thing there in the middle. So each of those sides, boop, boop, are genetically identical, and those are called sister chromatids. So this guy is just one chromosome, and then those two little copies of that same chromosome are sister chromatids. But remember, we have two copies of our chromosome. We have one from mom, one from dad. And so our copy from mom and our copy from dad are called homologous chromosomes. And so what this looks like is if my copy from mom is red, then I have my little duplicated chromosome there with my sister chromatids. And then if my copy from dad was blue, then I'd have like this. But these are both the same chromosome. So both of these are homologous chromosomes. They're the same chromosome, but one from mom, one from dad. So those are just some vocabulary words to get good at. The good thing about meiosis is there's some really similar mechanisms that we learned about in mitosis. But let me show you some pictures, it'll make more sense. But the idea about meiosis is that there's two phases. There's meiosis 1 and meiosis 2. And the prep for meiosis 1 and 2 is called interphase, similar to how we talked about in mitosis way back in Bio 430. In interphase, this is when we get chromosomes replicating. chromosomes replicate. They copy themselves. So notice in this picture, we have a green one and a purple one. And those are called homologous chromosomes, one from mom, one from dad. And this cell is diploid, which means it has two copies of chromosomes because there are two copies. After those chromosomes are replicated, notice now we have sister chromatid from mom, sister chromatid from dad. Again, still 2N because we have a green copy and a purple copy. In the process of meiosis 1, we separate those chromosomes. So notice that the green copy goes here and the purple copy goes there. So now we only have one copy of that chromosome per cell. So now these cells are considered 1N or haploid is that word. So that word haploid is important. Because we reduce the number of chromosomes between interphase and meiosis 1, this is usually called a reduction division. Reduction division meaning that we're reducing the total number of chromosomes from 2N, 2 copies, to N, 1 copy. So that's meiosis 1. But now we still have these sister chromatids. We need to separate them because we don't want two sister chromatids because it's the same information. And so then we'll take those sister chromatids and separate those. And then that's pretty much just like what we know from mitosis, where we take replicated chromosome and separate them. Separate those sister chromatids. This is often called an equational division. And it's the same steps as mitosis. But here we're still in one copy of chromosomes. The difference between meiosis 1 and meiosis 2 is that the difference between meiosis and mitosis is developing, or it is taking one cell. So mitosis is taking one 2N cell and making two identical 2N cells. Those guys are identical. With meiosis, we're taking one 2N cell and making four haploid or single N, 1N cells. The other thing is that these cells, these gametes are genetically different. Slightly different. Still containing all the same things that we need, all the right genes, but those traits that they carry that are ever so slightly different. So let's look at the phases of meiosis 1 and meiosis 2. A lot of these words I'm hoping will sound familiar to you. So first we'll start by talking about meiosis 1. That process of meiosis 1 starts with prophase 1. Prophase 1 looks really similar to mitosis in prophase. So notice what prophase 1, metaphase 1, anaphase, telophase 1. And then meiosis 2, it goes from prophase to telophase and cytokinesis, but they're all called 2 because it's meiosis 2. Okay, back up to the top. So back to meiosis 1, we have prophase. In prophase, it's really similar to mitosis. The other thing that happens is that we have like those spindle fibers still spit out from our centromeres, or from our centrioles rather, and then we'll still align those chromosomes along the metaphase plate, that's metaphase 1. Anaphase, we're still pulling apart those chromosomes. However, in anaphase, we're pulling apart homologous chromosomes. Notice that there's that green X and that purple X. They're still getting pulled apart as a unit. In telophase 1, we still start to kind of like reform that nuclear membrane and all of the things. And that gets us into meiosis 2. In meiosis 2, we'll start with prophase 2. Same thing still happens. Spindle fibers get spit out from our centrioles, and will still align along the metaphase plate. That's metaphase 2. Anaphase 2 is separating the sister chromatids, so notice this purple X is now one sister chromatid and two sister chromatids, and now they're being pulled in separate directions. Anaphase 2 is followed by telophase 2 and cytokinesis, which means that we'll pull those cells apart and separate them. And then we'll have 4 genetically unique daughter cells, and they're all haploid. I'm inserting these slides here, these next 2 slides, just to show you like the same information as before, but zoomed in. So hopefully it's easier to see on your devices when you go back. Okay. I really like this slide because it's a comparison between mitosis and meiosis. The thing about mitosis and meiosis here is that meiosis produces genetically unique daughter cells, while mitosis produces genetically the same, identical daughter cells. The reason for that genetic variability in meiosis is because of 2 processes. One of them are from this process called crossing over. Let's see, crossing over. Crossing over happens during prophase 1. That crossing over process, let me draw it here to the side. So let's use the green and purple from our picture. So if we have a green chromosome, and we have a purple chromosome, what happens during this period of crossing over is that it's kind of like, did you ever swap bits of your school lunch with your friends in elementary school? Like you'd trade your celery sticks for your friend's baby carrots. Like you still have a lunch, but it's different than it was before because you have the carrots and you gave away your celery. But you still have the same amount of food, right? And that's what crossing over is like. We trade little bits of chromosome, but you still maintain the same stuff. And so what happens in the period of crossing over is that maybe a little bit of purple chromosome will cross over onto green and then they'll kind of like trade. So what this could look like is this. So now this purple chromosome might have a little bit of green on it, like a little bit of green. And then our green chromosome could have a little bit of purple. Notice it's in like the same spot. You see what I mean? So that's crossing over. And that allows for a lot of genetic variability to happen because it's still the same chromosome, just with a little bit different stuff. So that increases genetic variation. And that's important when we only have one copy of chromosomes, right? The next thing that happens to increase genetic variation happens in metaphase. In metaphase 1, notice how chromosomes line up randomly at the metaphase plate. So notice how there's a purple one here, but a purple one on the other side. By the idea that our chromosomes randomly align along this metaphase plate also increases genetic variation. The reason for this is that when that metaphase 1 is completed, notice how this haploid cell has one purple from the top and one green from the bottom. And then the flip side is true of the other one. So that means that we each get a different, and then again, purple is from mom, green is from dad, or whatever. So that helps increase our genetic variation. This is the general process of meiosis. We're going to go over this more in the lab, but this is how our gametes are formed. So the end of this end process here are gametes. Sperms and eggs. And then sperm and eggs are formed through the process of meiosis by going through meiosis 1, then meiosis 2, with some slight variations that we'll talk about in a separate video.